Let's finish off cycle 2 with meiosis and recombination. We will touch on mitosis today, just so you know, but the focus will be on meiosis. Before we start lecture, just a couple of brief announcements. Simutext is a great platform to use for cycle 2, especially if you don't have a very strong background in mitosis and meiosis. Please understand that these topics are a prerequisite for this course. So between us, I'm not even supposed to be teaching you mitosis and meiosis. I should be getting right back, into advanced stuff with that topic. But what I've noticed is, everyone comes from different backgrounds in high school, and not everyone has the background they need. So rely on the textbook, rely on Simutext for something like this. But I will go over key concepts that I want you to know for mitosis and meiosis. And if you find that you are not able to keep up, or you're missing some things that you need to fill in, then fill in those gaps using those other resources. Please do complete the Test Your Knowledge questions. These are questions that are posted right after the lecture, and the solutions are there either in PDF format or video format. These, I would say, are exam level questions. My quiz questions, I wouldn't say are really reflective of exam level questions, because I tend to make them a little bit easier. I want you to get a good grade in quiz, but the midterm and final, it is what it is, so it's really important that you do these questions. I really think that will be really beneficial to you. The last thing I want to mention is the terminology that I'm going to use for not just this lecture, but subsequent lectures, teaching genetics, and specifically meiosis and non-disjunction events, looking at different syndromes, right? Looking at Down syndrome, Turner's, Klinefelter's, etc. It becomes complicated to keep the lecture gender neutral. I have tried to do it in the past, and I've done it successfully, but the students actually said, this is getting very confusing. The XY individual has an extra X. However, you're non-disjunctive, the XX goes here, then the XY individual, the XX. Like, I don't even know what you just said. Just say male, female. It's much easier, and I agree. So for the purposes of this course, I'm going to use male for XY, female for XX. I'm also going to say mom and dad. I also appreciate that, for some of you, you may not share genetic material with the individual that you call mom and dad. So this is not to disrespect anyone in the room. This is just, we have to get through these cycles in 15 minutes. And so it's hard to teach it. It's harder to learn it. So let's just keep it as simple as we can. Okay, what are the two things that we'll cover today? Number one, the fundamental principles of meiosis. I am going to compare meiosis to mitosis briefly, but my understanding is that you will know most of it on your own. The second thing we'll talk about are events that lead to genetic diversity. What is genetic diversity? If you want to know what genetic diversity is in a very simple way, just look in this room. Look how different we all look, right? We all have different phenotypes because of the genotype, and we all come from different backgrounds. We have different personalities. We inherently are good at certain things naturally, right? So this all comes into being diverse. So how is it that, we all come from a sperm and an egg? How is it that we have such diversity? We look so different. You could look very similar to your siblings, or you could look very different from your siblings, right? Unless, of course, you're monozygotic twins, but we're not talking about twins here. Why is that? You know why that is? Because of the events that occur during meiosis and after meiosis. So during meiosis, there are events that occur. What are those events? The key events. Number one, homologous recombination or crossover during prophase 1 and prophase 2. The second event that leads to this genetic diversity is independent assortment during metaphase 1 and metaphase 2. So homologous recombination or crossover. How many of you guys have heard of homologous recombination? Good, okay, so that's a term I will use as opposed to crossover. The mechanism of crossover is called homologous recombination. So that independent assortment. And the third thing that leads to genetic diversity, which comes after meiosis is complete, is the actual act of fertilization, which is random. So random fertilization. An egg and a sperm come together randomly. So you might be thinking, wait a second, like what about some organisms that know, even in humans, know who the mates are, right? Is that random? If it's already predetermined, sure, but you can't predict that this specific egg will be fertilized by this specific sperm, right? You can't say in January 2027, this specific egg will fertilize this specific sperm. It doesn't happen like that. It's random. So yes, you know what? We are going to say egg, sperm, ovaries, testes, sexual reproduction. And you know, I have friends that teach much younger grades, and they're like, oh, you use these terms like sperm and egg and sexual reproduction. Yeah, you know, I teach adults, so you know, we're all adults here, and I said most of my students are in medical sciences, health sciences, so yeah, they're good. All right, so those are the things that we're going to talk about. That's a summary of what we will discuss today. I want to go over the key differences between mitosis and meiosis. Key, the ones that I want you to know. Mitosis, it is a one cell division event. So one division event right here, one cell division. It produces two daughter cells. They're genetically identical, and the ploidy doesn't change. The ploidy remains the same. Don't worry so much about the term ploidy for this lecture, but you definitely have to know it for next lecture. So what does ploidy mean? Ploidy is how many unique sets of chromosomes an organism has. So we have two sets of chromosomes, two sets of unique ones, one from one parent, one from the other, right? So we are diploid. We are 2N. We are 2N organism, which is a diploid. So ploidy remains the same throughout mitosis. What's our N value? Does anyone know what their N value is? Yes, 23, excellent. We have 23 unique chromosomes. So N equals 23, so we have 23 unique ones, times 2 is 46. So we are 2N equals 46 organisms. So all these organisms can be characterized mathematically. That's really what the characterizing genome means. Meiosis is a two-division event. First division here, second division here. It produces 4 cells, so the end product are 4 cells. They're not genetically identical, and the ploidy does change. Why does a ploidy have to change? Because these cells at the bottom are gametes. They're haploid. Because one gamete comes together with another gamete, for example, an egg and a sperm, and they produce the 2N zygote. That 2N zygote then now multiplies, right? G1SG2M, G1SG2M differentiates into an entire human being. So you start off with 2N, but then you end up with N. The next slide is a clicker question. I'm not sure if clicker's working, but if it's not, we'll do raise of hands, and we will just go from there. If you cannot answer this question correctly, I don't want you to freak out. I'm not expecting that everybody will know this. This is not high school review. I just want to have a sense of how much the class knows and how comfortable you are with this topic. So let's see if it works. If clicker doesn't work, then we will. It doesn't look like it's working. Yeah, sorry guys. The Wi-Fi's bad, right? Okay. You know what, if it works, it works. If not, we're gonna do a raise of hand. Just focus on the question. And please commit to an answer in your head. Commit it. On the exam, you can't go, oh, I'm just gonna wait for the answer. No one's gonna come with the answer for you. So learn, fire those neurons in your brain to commit. And if this was a question on the exam, what answer would you choose ultimately if time's running out? Now on an exam and a quiz, you will get much more time than this, but we'll just stop it here. How many say A, B, C, D? Okay, I want to hear from someone that said D. Yes? D means that 2 is out.
During meiosis 1, the chromosomes are separated differently than mitosis. Meiosis 1 produces haploid cells, whereas meiosis 2 doesn't change the...
Right, good. That is the correct answer. I wanted to know why 2 was excluded. Okay, so the correct answer, guys, is actually D. D as in dad, D. 2 is out. 2 is incorrect because meiosis 1 is the reductional phase. It's in meiosis 1 that you see it going from diploid to haploid. Haploids are made right away during meiosis. So let's go over this. Whenever I take a multiple choice question and go over it, that's a good sign. Just understand that there could be exam questions coming out of this. Take every question in the future, practice questions, and do what I'm doing with it, okay? Watch what I'm doing with it. Okay, statement number 1. Do you know why meiosis 1 is not identical to mitosis? Okay, so let's go over this. Give me some differences between the two, meiosis 1 and mitosis. Okay, the answer is mitosis is 1-cell division, meiosis is 2-cell division, but the question is what's the difference between meiosis 1? Meiosis 1 is a 1-cell, right, division. Anyone else? Yes. Excellent. There's homologous recombination in meiosis 1. There was no homologous recombination during mitosis. Excellent. That's one really big one. What's another big one? Yes. Okay, yes. So mitosis creates genetically identical. Meiosis 1 doesn't. Okay, yes.
Homologous pairs are separated during meiosis 1, whereas they aren't.
Very good. During meiosis 1, homologous pairs of chromosomes are separated, not sister chromatids. There's no separation of sister chromatids. If you see an image on an exam with homologous pairs separating, and I'm saying this is anaphase, do not pick anaphase of mitosis, pick anaphase 1 or anaphase 2, right? Would that be anaphase 2 or anaphase 1 out of curiosity? Yes. Anaphase 1. So please go to the textbook, guys, go to Simutex, understand this concept. It will be on the exam. So understand how the separation occurs and what's happening where. What's another thing? During meiosis 1, the ploidy changes from 2N to N. During mitosis, there's no change in ploidy. If anything, guys, if mitosis is similar to anything, it is more similar to meiosis 2 than it is to meiosis 1. Okay? If there's going to be more similarity, hands down with meiosis 2. We already went over too, meiosis 1 produces the haploid cells, whereas meiosis 2 also does produce haploid cells, but it goes from haploid to haploid. It stays the same. Number 3, homologous recombination occurs between 2 identical sister chromatids. How would you sentence this correctly? How would you write that so that that sentence is correct? Yes? Homologous recombination occurs between 2 homologous pairs. 1 homologous pair, yes, it does occur between 1 homologous pair. I want it in terms of sister chromatids. Yes? Very good. Between non-sister chromatids. Non-sister chromatids. The products of meiosis are non-identical gametes. Yes. If the gametes were identical, then we wouldn't have the genetic diversity that we do. Excellent. Okay, so now let's go back to this slide. So let's fill this in. 2N, it stays 2N all throughout. All these cells are 2N. Here, this is 2N, but after cell division, this is the reductive phase, and so this becomes N right away, and then all of these are N. They have to be N because 2N come together to make the diploid zygote. Another thing that I want to go over, again, if this is review for you, I appreciate your patience, but if this is not, then I think this will be very helpful. Where does meiosis occur where and when in the body, and when does and where does mitosis occur. So females have ovaries, males have testes, and within these reproductive organs, you have these cells called germ cells, G-E-R-M, and these are 2N. They're going to undergo a process. So in the ovaries, you'll produce the egg, testes, the sperm. They're going to undergo a process, which you don't have to know, like what it is, how exactly it's working, but in testes it's called spermatogenesis, in ovaries it's called oogenesis, but ultimately what's happening is meiosis 1 and meiosis 2 in both, meiosis 1 and meiosis 2, to produce the haploid gametes. N means you have one unique set of chromosomes, one unique set. Unique set means that each chromosome is a different chromosome. That's one set, right? So one set is coming from mom, one set from dad. That produces your 2N one-cell embryo called a zygote. Now this zygote will divide via mitosis the entire time. There's no meiosis going on. Mitosis the entire time, G1S, G2M, G1S, G2M, as you learned last class, and it will differentiate and will become an entire organism. So this is how mitosis, meiosis works in terms of differences and when and where it happens. The only time you see just the beginning of meiosis happening during embryogenesis is in the ovary. You see just the beginning of that germ cell entering meiosis, and those cells then arrest in prophase 1. Then meiosis is completed in the fallopian tube after puberty every month. Now let's go into details about meiosis 1 and meiosis 2, and then we'll talk about aneuploidy. Meiosis 1 is reductional, meaning that it goes from 2N to N, as you can see here. To understand how this happens, there are a few things I want to go over. First thing is this. I want to go over the definition of a chromosome, what a chromosome is, what sister chromatids are, and what chromatin is. Would that be helpful, or you guys already know this stuff? Okay, forget it, Nikki, just don't waste our time. Go over it? Yes? Okay. This is a chromosome. The center doesn't always have to be in the middle. Okay, it has a long arm and a short arm chromosomes. Don't worry about all that. Just worry that there's a centromere. It can be higher, it can be lower, it just depends, but I drew it in the center just for simplicity. This is one chromosome. That's one chromosome. When the DNA duplicates during S phase, and then you get the DNA condensing and whatnot during prophase, you get sister chromatids. These are sister chromatids. However, this is still one chromosome. So you're like, but there are two strands of DNA. Isn't that two chromosomes? No, it's still one chromosome. Because the number of chromosomes is determined by the number of centromeres. If you know that, you will never get a question wrong on the exam. These two are sister chromatids, sister chromatids. So for example, if this is chromosome number 10, right, you have chromosome number 10 from one parent, you also have chromosome number 10 from the other parent. These are homologous pairs. These are homologous. These are also sister chromatids, and this is also one chromosome. If you compare this one and this one, these are non-sister chromatids. These are non-sister chromatids. If you have a cell with a nucleus and you see this mesh that looks like this, that is chromatin. So you have like histones, DNA, histones, DNA, histones, DNA, because it wraps it up with histones, right? That's what your DNA actually looks like in the nucleus. So is that clear to everyone? What's what? All right. When you understand the definition of a chromosome, then you can follow ploidy. Otherwise, it's difficult to follow ploidy. Now look over here. You have meiosis 2. The example that's given here, sorry. I meant to write all that stuff in this slide, but it doesn't matter. You understand the definition of a chromosome. If you look over here, these are homologous pairs. Only two pairs are shown. But let's just say this is chromosome number 10, and this is chromosome number 5, right? You have two sets shown here, two sets. A set is 10 and a 5, 10 and a 5. A set is not both 10s, one set, both 10, both 5s, one set. A 10 and a 5, 10. The set has to be unique chromosomes in there. You can see crossover or homologous recombination occurring. Then these line up in the metaphase plate. The lineup is going to be independent of each other. This is where you get independent assortment. So for example, if chromosome 10 has this allele, A, big A in the top one and little a, right, heterozygous, let's just say the heterozygous here. This chromosome is going to line up independently of this one, meaning it's not going to see the big A on top and go, oh, I also got to put my big B on top. No. If it does, it does. If it doesn't, it doesn't. So it can line up like this, or it can also line up like this. Okay? So it's independent. This is why, and this is just two chromosomes that are shown. Imagine all of them lined up independently of one another. This is why you have this genetic diversity. What's the probability that the lineup will always be exactly the same, right? And the crossover will happen exactly where it's supposed to, like all the time. This is why we have it. So these are the two big events, homologous recombination and independent assortment. Now these homologous pairs will separate. There's no sister chromatids separating here. Once they separate, look what you have now. How many sets do you have? You have one set. This is chromosome 10. This is chromosome 5. So you might be saying, okay, so did I go from 4 chromosomes here in 10 to 2 chromosomes? No, you went from 2 chromosomes here to 1 chromosome. Count the centromeres. The copy number has changed. That's a different thing we'll learn next class. And the chromosome number has changed, but in the next slide you'll see that it's going to be a little bit different in meiosis 2. So let's go to meiosis 2. You're left with this, right, from the previous slide. So this is now, again, going to line up, and the sister chromatids are now going to separate in meiosis 2, and you end up with these haploid cells. Because, see, look at this. Even though you're saying, wait a second, I have 1 centromere here, that's 1 chromosome, and I have 1 centromere here. So yes, the ploidy does not change. It's still N. That's why. You count the centromeres. However, you do have 2 copies here. You only have 1 copy. You can only have 1 copy in the haploid because the other parent will bring the other copy. All right, so this is how you get the production of these haploid gametes. N, 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 and it starts off with N. This cell over here in the beginning is not 2N. So, details of homologous recombination. Here's an example. If we say that, for example, mom is in the red here and dad's is in blue, then these are homologous pairs. Mom has, happens to have capital A, B, and C, meaning like, doesn't have to be the dominant one, but let's just say it's dominant. And this is the recessive one, so you have a heterozygote that could be formed when crossover occurs. And so when you do those Punnett squares and you go A, little a, big A, this is what's happening, right? It's during crossover. That's how you get those possible progeny. The point where it crosses over is called chiasma. The plural of chiasma is chiasmata. You can have multiple chiasmata occurring during, that crossover event that occurs between homologous pairs. Homologous pairs do not sit side by side because this is a 2D picture. They actually are on top of one another, forming a tetrad. And this is the reason why all sister chromatids have the ability to undergo homologous recombination. Not all of them will, but they all have the ability to do it. How many chiasmata do you see? Anywhere from 1 to 4. Okay, and this is random as well. Here you can see, once anaphase 2 is done and all the sister chromatids are separated, when you get this random segregation into the cells, you're going to see that each one is going to receive a different version, right? This one has all ABC big. This one has AB small, C big, AB big, little c, little, little, little. So all of these gametes are going to have a different version or different allele of these genes. These genes represent the same thing. They just have different versions of them, right? So A is the same as little a in terms of the function of that gene. It's just a different version. What does that mean? The sequence in the DNA is different. Therefore, it can be, it just depends on what that version is. Is that version doing something completely different? Is the version doing something very similar? So that's how you get your traits. So when we talk about chiasmata, a very common topic that comes into play here are linked genes. And just an FYI, students that get a 98 on the exam, this is the question that they actually mess up. So this is, and I don't know what it is. I think because linked genes, it's a very, very easy concept. But when you take this concept and couple it with something else, that's where it starts to get a little more complicated. So just please pay attention here. Linked genes simply means that if genes are linked, then they're more likely to be inherited together. So what does linked mean? Linked means that they're close to one another. So like literally in physical proximity to one another. So here you have a chromosome with gene A, B, and C. These are the locations of these genes. And you can see that gene A and B are very close together. However, they're distant from gene C. So what's the probability, guys? It is all probability. This is not like, well, there's a 0% chance or a 100% chance. It's just probability. What is the probability that the chiasmata will always occur between A and B? Less, right? Less than the chiasmata occurring in this distance. It's like, for example, if there's a big styrofoam chromosome here, and I take a bow and arrow, and I have to try to hit this chromosome, the chances that I'm going to keep hitting there, because, well, first of all, I'm not really good at using bow and arrow, but the point is that the probability that I will hit somewhere here is much higher, right? So that's exactly the same concept. Is the probability that it'll keep hitting there is very rare compared to here. It's just relative. This means that if there's no chiasmata occurring here, then these genes will not be separated, right? During homologous recombination, they'll stick together. So take this concept, and then you're going to have to couple it with something else. When we ask you, tell me which gametes will be formed. Give me a probability of the gamete formation. I will not tell you the genes are linked. I will simply have the genes showing you where they are, right, in the loci. And to say they're here, here, and here, you have to know that, wait a second, these two are very close to one another. I think they're linked. The probability they'll be inherited together is higher. So you have to just get that one step further, which I think gets missed. But I'm kind of giving you the hint of how to answer a question like that. So if you go in your Simutext platform, then you can simply just rearrange the genes as you wish, and then you can give it, results of however many pairs of chromosomes you want, and it will do a random crossover, and it will tell you what the probability is that these genes will be inherited together or not. So genes A and C, the probability that they will be inherited together is 53%. A and B, 93%, which makes sense, right? Because they're so close to one another. So we talked about homologous recombination, which was the first event that leads to genetic diversity. The second one is independent assortment. We already touched on that. I told you what independent assortment is, but I just want you to see it this way too, using this example, which is a lot more clear. I mean, the different gametes are right there, right in front of you. So you can see the different genotypes that's produced due to independent assortment. This example is actually that of Gregor Mendel using the peas. How many of you guys know Gregor Mendel with the peas cross that he did? Yeah, so you have the big R for round, little r for shriveled, big Y for green peas, and little y for yellow peas, right? If you look at the way these two are placed, these homologous pairs, then you see the big R here and the little r here, right? So is big Y and little y also going to align exactly the way R is? Maybe. Or it may align this way. So it can align this way or this way. So if it aligns this way, look at the genotypes of the gamete. If it aligns this way, look at the genotype of the gametes. they are different. All of them are different. And so just again, this is adding to the genetic diversity depending on how these chromosomes align. They align independent of each other. The last event is random fertilization. This is more intuitive to everybody. Random sperm, random egg coming together. And both of these have already come from randomization as well, right? They've come from homologous recombination, they've come from independent assortment, and now there's an addition added to that, which is the fertilization itself is random. So if you put all of this together, do you guys appreciate how it is that we have such a large population in the world and we all look so different? What happens when there's a problem in meiosis 1 and meiosis 2? What happens then? We touched on this last lecture, and we looked at aneuploidy and the chance of it occurring during metaphase. And we talked about the mitotic spindle checkpoint being a really important part of the cell cycle to make sure that things are checked so that this doesn't happen. How does aneuploidy happen? It happens when there is a non-disjunction event. Non-disjunction means when the sister chromatids are not separated properly or the homologous pairs are not separated properly, and an extra chromosome ends up in one cell and one cell gets one less chromosome. That's a non-disjunction event, improper separation of chromosomes. So here you can see the error that occurred was in meiosis 1, right? One of these was supposed to be in here. So the cell is going to get one small one, one big one. The way I'm going to tell you that chromosomes are different, I may not number them often, I will make them different sizes, and that's how you know that that's a different chromosome. So here, there are different sizes. So one will have a small one, one will have a big one. Don't put two bigs in one and then two smalls in the other. That's not proper segregation. And then you can see here that once these cells divide, right, you have an extra one in this, one less in this. So this gives you n plus 1, and these two give you n minus 1. N is a normal number, right? Our normal number is 23. And so in this case you have 24. In this case you have 22. This would be an example of Down syndrome. This will be an example of Patel. This will be an example of Edwards syndrome. So this is how it happens, a non-disjunction event during meiosis 1. This is pretty detrimental, right? If you had a non-disjunction event during meiosis 1, all of the gametes are affected. All of them. So, yeah. But if you get a non-disjunction event in meiosis 2, you have a better outcome because some gametes end up being normal and some are affected. All right, how am I going to ask you a question on the exam like this? I'm going to ask you the following. Here is a syndrome. This is the genetic makeup of this individual. Where did the error occur? Was it in mom? Was it in dad, first of all, or both? It could be either or. And when? During meiosis 1 or meiosis 2? You have to work that out. It is one of the most complicated questions, at least in my cycles. Yes? Yes. So we'll talk about what happens. No, you don't eventually die. So like, for example, there are, we will look at when you get less chromosomes, you're still fine, okay? Okay, so now, I'm also going to tell you that the error is a single partitioning error. So in other words, you don't have to make an error in all these permutations. Okay, so where's the error happening? Is it in meiosis 1 and also in meiosis 2? Is it in meiosis 1 in the dad, meiosis 2 in the mom? That's ridiculous. I mean, you just can't solve a question like that. You make there's just one error that has happened, one, throughout the entire process, including mom and dad together, one error in partitioning. Where could that error be? And that would be a multiple question. So I have put up videos online for you to see how to solve a problem like that. Do they test your knowledge? There's a question like that. So make yourself familiar with this. This will be on the exam. What happens if there's an error in meiosis 2? In other words, if you get non-disjunction in meiosis 2. Meiosis 1 goes fine. Now the non-disjunction is in meiosis 2. In this cell here, a single error. Don't make the error in both of them because then the outcome will change, right? So let's just keep the course simple for now, and we just do the error here, and there you go. This is n plus 1, n minus 1, and an n. So meiosis 2 error does have a better outcome because you have, half your gametes looking normal. So just, again, it just depends on which one is the one that's going to get fertilized. Down syndrome, as we mentioned, is an example of the outcome of non-disjunction. And the reason I'm talking about Down syndrome is because this is the best outcome you can get from an autosomal aneuploidy. From autosomal. Autosomal means like your 1 to 22 chromosomes, 1 to 22, and then you have your sex chromosomes. Sex chromosome aneuploidy has a much better outcome. So here's a karyotype. What is a karyotype? You're going to see lots of karyotypes in my exam, so be familiar with the karyotype. A karyotype is when, this is a picture taken by a scientist. It's developed by the scientist, and the picture is taken using a camera. And the way it works is they look at the cell during metaphase, and then they gather up all the chromosomes. They line them up from largest chromosome to smallest chromosome, and they put the sex chromosomes in the end, and they take a picture. And that is a karyotype of the organism. The karyotype of almost all organisms are published. If you want to have a look and see what the karyotype looks like, it tells you a lot of information about the genome of that species. So here you can see, you can also detect syndromes using the karyotype. You can see that you can see 3 copies of chromosome 21. That's how you know that this individual has Down syndrome. 3 copies right there. Here's a graph of the mother's age on the x-axis and the incidence of Down syndrome on the y-axis. You can see there's a marked increase after the age of 35 in the incidence of Down syndrome, and that is because of the fact that the eggs are not being regenerated, right? The eggs that the individual is born with. And guess what's happening over time? That egg is accumulating mutations. As you live life, you take in exogenous sources and you're living life, right? I mean, your DNA is changing. And you will learn more about how much your DNA changes throughout your life. We'll talk about that. And so there's a change. So if you're going to get mutations and changes in your genome in cells of your body, then why would your germ cells be an exception, right? Why would the germ cells be, oh, we're not going to touch the germ cells, we're good, we're going to keep going with your heart cells and liver, no, germ cells are safe. No, you can't help it, you're living life. And so, yeah, as you age, you're just accumulating more mutations. That's one factor. The second thing is, there's a lot of pressure on that egg, because that egg constitutes the bulk of the zygote, right? The sperm is there, you get the DNA from the sperm and some parts of the sperm, but for the most part, that egg can't just say, well, you know what, I maybe can't do my function in the heart so well, I maybe can't do my function in the liver so well. It's not a liver cell, it's not a lung cell, it's an egg. It needs to become an entire individual. So you need to make sure that everything's perfect in there, right? Go through all of this path of embryogenesis. I mean, it's not a joke. So there's a lot of pressure there for everything to be perfect. If you look at a fluorescently labeled egg of someone in their 20s as opposed to someone in their 40s, there's a marked difference in just the way the spindles are labeled, just the way the chromosomes are aligned. The alignment is not proper. You can just tell that it is doomed for non-disjunction. Okay? Trisomy 13 and trisomy 18 give viable outcomes. In other words, the baby is born, but the outcome is poor. Viable, but outcome is poor. Most of them that are affected with these types of trisomy, they die before birth, or they die within the first, couple months of life. So a poor survival rate for that. But so Down syndrome has the best outcome. For autosomal non-disjunctions, but for sex chromosome non-disjunction events, that do also occur, the outcome is much better. So here you have Turner syndrome, Klinefelter syndrome, triple X syndrome, right? Turner syndrome, 1 less chromosome. So that was your question. Remember you said, what happens, is it viable? Is this embryo viable if there's 1 less? Here's an example. So if there's 1 less X, then it's viable. Y alone is not viable. You need the X to survive. The embryo needs the X, period. You know, X has a lot of chromosomes on it, right? X is a much bigger chromosome. Y is a much smaller chromosome. It doesn't have a lot of those real survival genes. So you can actually survive without the Y, right? I mean, there are many XX individuals, but I'm just saying, like, even if you're, you know, if you don't have the Y, you're fine. But if you don't have the X, then it's not viable. Turner syndrome X, Klinefelter's is XXY. These are males that have an extra X. It says 1 in 2,000. I think this is older data. From my recent reading, it's 1 in 650 men that are XXY. Many, many, many men go through life and they don't even know that they're XXY. So it just depends on your physical characteristics, your physiological characteristics, like what's happening inside your body. It ranges from severe to literally nothing, and it just depends on the individual. Most of the individuals that see symptoms of this type of syndrome have to do with fertility. So there's reduced fertility, reduced testosterone, reduced hormones, or just complete infertility. So that's where they see most of these characteristics that it has to do with fertility. But yeah, for the most part, very viable. You know, there's a lot of genetic counseling. A lot of families go for genetic counseling because they have a specific disease in their family that they want to look at. And you know, genetic counseling is a really big field if you're interested in going in there. And now it's changing rapidly, right? Because of what's happening with how we can detect things in the genome. So when they go for this genetic testing, that's when they find out that they have different combinations of chromosomes. So, and sometimes that explains their symptoms, and sometimes it doesn't because they don't have it. Here's a karyotype of the supermale, which is the double Y chromosome, XYY. The way you denote it is 47, meaning that you have an extra chromosome, comma, XYY. And this is the karyotype. Yes?
Do you want us to know the notation for certain?
Yes, so please do know the notation. Like, understand if it says 47, comma, XYY, then you know that this is, don't memorize. Which one is Turner's, which one is supermale, which one is Klinefelter's. I would tell you on the exam, you know, this is what it's called. We do no memorization, but just understand how to read a karyotype, what it means. We'll cover more of that next class. What I want you to do now for the next 3 slides, and please stay seated until the 20 mark, because what we're gonna do is I'm gonna tell you what you need to know. It just makes your life a lot easier, because I don't want you to throw you in the textbook and go, read it, read all of it. You're frantically taking notes, and only to find out that you didn't even have to know all this information. I want you to know the life cycle of the animal, plant and fungi and algae. I want you to know that. And what I want you to focus on is the following. The reason I'm telling you to read it in the textbook, because this concept is absolutely straightforward. If I were to stand here and give you a lecture on this, all I would be doing is regurgitating the textbook. So it doesn't make sense for you to come here for that. So go to the textbook, read it. It's really well written. You know, you read something, you're like, that was really well explained. This is one of the concepts. So here you have the 2N animal as an example. Meiosis is what produces the gametes. Gametes arise by meiosis, right? The gametes are here. And then fertilization occurs, and then the zygote, which is 2N, it divides by mitosis. Okay, so this is the diploid phase, and it has a haploid phase. This may be very intuitive to you, right? Because we've been covering animals and humans, and you know, so we know. What may not be as intuitive is this one here. How many of you guys have done a lot of plant studies in your high school? Just curious. The plant people, raise your hand high. Awesome, yeah. Plant research can change the world, right? You guys know that. So plants have alternating generations. They can live in the diploid phase, or they can live in the haploid phase. So look at this carefully. Gametes arise by mitosis, not meiosis. Gametes arise by mitosis. Spores are formed by meiosis. Do not mix all of this stuff up on an exam. You will get the question wrong, so pay attention, please. When this gets coupled with genome characteristics, that's when things start to go a little bit more complicated, but that's how I will test you. I will test you by coupling cycle 2 and cycle 3 together. all test questions, or majority of them, will be coupled, right? So learn how to integrate one concept and then the other. You will have plenty of practice. Here you have the sporophyte, which is 2N. Within the sporophyte, there's a region that is going to undergo meiosis. And through meiosis, spores are produced. This, my friends, is where the genetic diversity occurs. The spores are genetically different. Then the spore divides by mitosis to produce the gametophyte. Within the gametophyte, there's further mitosis, which is, this is very generic, like there's more details here, but just this is all you need to know. It forms the gametes. The gametes then fertilize to make the 2N zygote. The zygote divides by mitosis. So understand this. Last one is fungi and algae. Gametes arise by mitosis. The zygote divides by meiosis. So I know this all sounds confusing, but just read the textbook, you will get it. So look at this as well. It has a haploid phase and a diploid phase. The diploid phase is very short. This lives as haploid. You know when you open your fridge sometimes and you take out something that you forgot and has fungus in it? Yeah, this is it. It's sitting there in the haploid phase in your fridge. All right, so on that note, this is the last lecture for this cycle. I will see you guys next week. Yes, thanks. Thank you. All right.